So we can proceed. So we're on step number 5. We've looked at step number 4, which is basically the cleavage of fructose 1,6-bisphosphate into 2,3 carbon molecules, which is glycerolaldehyde 3-phosphate and dihydroxyacetone phosphate. The breakdown of fructose 1,6-bisphosphate is catalyzed by the enzyme aldolase. So let's go to step number 5. So step number 5 is a very easy step because this is basically the isomerization of dihydroxyacetone phosphate into glycerol-3-phosphate. This is because glycerol-3-phosphate is the only molecule which can be used for the rate of glycolysis. So therefore, dihydroxyacetone phosphate is converted rapidly into this is converted via a reversible reaction into glycerol-3-phosphate. The enzyme that is involved for this isomerization is triose phosphate isomer. So this enzyme basically moves. I really don't know what I can do about that because I think I've tried everything I can. So if my voice again is there, going and coming, I don't know what I will do. So I was saying that triose phosphate isomerase basically just moves the phosphate group from the ketone form to the aldose form. So in this case, you have CH2 so you have CH2 OH. Yeah, hydroxyacetone, by the way, is a ketone group, has a ketone group rather, 2 minus, that is a 3, that is a 2. Then we are saying that is converted to glycerol-3-phosphate. So all you've done is to move the phosphoryl group from a ketone. From our 12 lectures, we looked at the structures of a ketose and an aldose. So an aldose has got an aldehyde group, whilst a ketose has a ketone group. So the functional group is there, and the functional group is there for an aldose. Let's go to step number 6. So step number 6. Step number 6 involves the conversion of glycerol 3-phosphate to 1,3-bisphosphoglycerate. So 1,3-bisphosphoglycerate. So the reaction is catalyzed by the enzyme glycerol aldehyde 3-phosphate dehydrogenase. So it's catalyzed by the enzyme glycerol aldehyde 3-phosphate dehydrogenase. Within this reaction, the reaction also uses NAD+, which is a reducing equivalent to form NADH. So the reaction proceeds in this manner. So you have the glycerol aldehyde phosphate, or H and H. Carbon number 3, we have the phosphoryl group minus. Then here, what we are adding is NAD plus and an inorganic phosphate molecule. And then what we are getting is NAD plus so NADH. So you find sometimes we write it as plus H plus, like that. The whole reaction is a reversible reaction catalyzed by glycerol 3-phosphate dehydrogenase. Like that. What we are producing is 1,3-bisphosphoglycerate. So at carbon number 1 and carbon number 3, we have phosphoryl groups. So PO3 minus. Then we have OH. We have an H there. And we have CH2 or PO3 2 minus. So this is 1,3-bisphosphoglycerate. And this one is glycerol 3-phosphate. So basically we're converting the glycerol 3-phosphate to 1,3-bisphosphoglycerate. The difference here is that the bisphosphoglycerate has got 2 phosphoryl groups. Let's go to step number 7. So step 7 is just the conversion of 1,3-phosphoglycerate to 3-phosphoglycerate. Now, this is the first step where we are synthesizing an ATP molecule. So the reaction results in the conversion of 1,3-bisphosphoglycerate to 3-phosphoglycerate. The process is catalyzed by the enzyme phosphoglycerate kinase. So this enzyme catalyzes the transfer of the phosphoryl group from 1,3-bisphosphoglycerate, 1,3-bisphosphoglycerate, which I've abbreviated as 1,3-BPG, to generate ATP. So this is the first step of where ATP is formed. So just put that down. So we're saying 1,3 is phosphoglycerate to form 3-phosphoglycerate. All right, the structures. So we have 4, 3, 2 minus. So we have a COH there, a hydrogen, and we have CH2OPO3, 2 minus for the phosphate. This whole reaction is a reversible reaction catalyzed by phosphoglycerate kinase. And then we have 3-phosphoglycerate. CH2O3P minus 2. And then we would have formed a molecule of ATP. So this is the first step that results in the production of ATP. We can go to step number 8. So step 8 is basically the conversion of 3-phosphoglycerate So this is converted to 2-phosphoglycerate by the enzyme phosphoglycerate mutase. So the enzyme is phosphoglycerate mutase. Mutase, like that. So that's the enzyme that converts the 3-phosphoglycerate. The structures for that. So we start with the 3-phosphoglycerate, which we had in step 7, which we had produced in step 7. So this is 3-phosphoglycerate or 3-PG in the presence of phosphoglycerate mutase as an enzyme. Then we are forming 2-phosphoglycerates. So this is a reaction in which you are just changing the position of the glycerate. I mean the phosphate group. So that is 2-phosphoglycerate. Step number 9. We can go to step number 9. Step number 9 is a dehydration reaction. So this is dehydration of 2-phosphoglycerate to form phosphoenolpyrrole. Abbreviated as PEP. This reaction converts the low energy phosphor-ester bond of 2-phosphoglycerate into a high energy profile bond. So basically, the reaction is catalyzed by the enzyme enolase. So catalyzed by the enzyme enolase. The structures associated with that. So we start by drawing the structure for 2-phosphoglycerate. Hydrogen there, CH2OH. Again, the reaction is reversible. So you have enolase as the enzyme. We have said that this is a dehydration reaction, so we are losing water. And then we are forming phosphoenolpyrrole. So we have that, carbon number 2 minus. And then we have put basically a double bond between carbon number 2 and carbon number 3. So this is 2-phosphoglycerate. And this side we have phosphoenolpyrrole, abbreviated as PEP. Step number 10, which is the final step or the last reaction. This results in the transfer. In step number 10, there is a transfer of the phosphoryl group phosphoenolpyruvate to ADP to form ATP and pyruvate. The reaction is catalyzed by the enzyme pyruvate kinase. So we can write back the structure for phosphoenolpyruvate. So we have the carbonyl carbon, the carboxylic end, PO3 minus. Then you have the double bond there. So this is phosphoenolpyruvate. And then here we are adding ATP plus and a hydrogen ion. And then we are getting ATP under the catalysis of pyruvate kinase. And then we are producing the end product, which is pyruvate. So here we have pyruvate and the TEP, which is the phosphoenolpyruvate. So phosphoenolpyruvate, and then we are getting pyruvate this time. Okay, so these are the 10 steps of glycolysis. From step number 5, if you recall, step number 5, we said, or we can start from step number 4. So step number 4, we said we break down the fructose 1,6 bisphosphate into 2,3 carbon molecules. So since a glucose molecule has got 6 carbons, when we break the 6 carbon into 2,3 carbon molecules, it means that from step number 5, the reaction will repeat twice, since we have 2 molecules of the same thing. So in this case, since the dihydroxyacetone phosphate is converted also to glycerol 3 phosphate, means that we have 2 molecules of glycerol 3 phosphate. What this entails is that the whole glycolytic process runs twice or 2 times for every 1 molecule of glucose. Because glucose has 6 carbon atoms, and therefore when it's split into 2, it means that it will run twice from step number 5. What this means is that since we are producing ATP, the first ATP that we have produced in this reaction is step number 7. So it means that step number 7 will run twice, meaning that we are producing 2 ATPs. And also step number 10 will run twice, meaning that we would have produced 2 ATPs. So in terms of energetics, we have invested 2 ATPs in the investment stage, step number 1, where we added ATP there. Step number 3, where we added ATP there. So we actually invested 2 ATP molecules, but we are investing 4 ATP. Produced, meaning that for every cycle of glycolysis, So the net gain in a cell of glycolysis is 2 ATP. The other thing that we want to make mention is that the formation of ATP is due to two types of phosphorylation. So in glycolysis, we have two types of phosphorylation. So we are calling it substrate level phosphorylation. So substrate level phosphorylation is the production of ATP or we can say that this involves transfer of a phosphate from a sugar phosphate intermediate to ATP to form ATP. Okay, so we'll see another type of oxidation, rather substrate level, or rather another type of phosphorylation, which involves oxidative phosphorylation. And that one involves the transfer of electrons. So those are the 10 steps of glycolysis that you need to be aware of. The other aspects of glycolysis is basically the control of glycolysis. So quickly, I'll just run through that. So the control of glycolysis is dependent on basically the three steps which are irreversible. Step number one, if you check, is irreversible, which is catalyzed by hexokinase. Step number 3 is also irreversible, which is catalyzed by phosphofructokinase. And step number 10, which is catalyzed by pyruvate kinase. So you need to know how glycolysis is controlled based on those three enzymes. What is it that inhibits their activity, and what is it that activates their activity? Mostly, you will find that most of these enzymes will be inhibited with excess ATP. So if there is a lot of ATP in the cell, these enzymes are going to be inhibited so that no more ATP is formed. So I think we can end here for today. I will accept questions if there are questions.